Hi class, in this recording we're going to focus on the three major buffering systems, chemical buffer systems in the human body. The bicarbonate, the phosphate, and the protein buffering systems. So let's dive into it. Let's look at the bicarbonate buffering system. It all depends on carbonic acid and bicarbonate ions. So this is a chemical reaction that you had to memorize for the respiratory system. As we have carbon dioxide being dumped into the bloodstream, it can be turned into carbonic acid, bicarbonic anhydrase, in the erythrocytes, and then it gets turned into, dissociates to bicarbonate ions and hydrogen ions, which then are going to be moving around in the body. And that's how we move most of the carbon dioxide in our bloodstream. We actually convert it to a bicarbonate ion and dissolve it in our blood plasma. Because this reaction can head both directions, we have these double arrows right here. That's a big clue that it's a reversible reaction. And the thing that determines the direction is, in a word, or in a term, Les Chatelaires Principle, for those of you who've taken more chemistry classes. And the idea behind Les Chatelaires Principle is that if you have lots of stuff on a high concentration of chemicals on one side of the reaction proceeds away from to produce the thing that you have a low concentration of. Or if you have a high concentration on the other side of the reaction, the chemical reaction proceeds the other direction. So as we're looking at this particular buffering reaction, it's also going to be, Le Chatelaire Principle helps us predict how this chemical reaction responds to a change in pH. So let's say we take this chemical reaction and we expose it to a base. A basic chemical compound is going to remove hydrogen ions from solution. So we start to suck up hydrogen ions with the base. And that lowers the concentration on this side of the chemical reaction relative to the left-hand side and drives the reaction this way, thereby releasing more hydrogen ions. And on the flip side, let's say we're introducing extra acid to the chemical equation. If we're introducing extra acid to the chemical equation, that's going to lower our overall pH and increase the concentration of hydrogen ions. And as we release more hydrogen ions into solution, the concentration of hydrogen ions goes up. And because the concentration on this side of the reaction becomes higher, the chemical reaction is driven this direction. So either way, we are going to be able to predict the chemical reaction based on the relative concentrations of hydrogen ions. If we're adding hydrogen ions, we compensate by binding them and removing them from solution. If we are removing hydrogen ions from the solution, we compensate by releasing more into solution. That's how this buffering works. As we look at the bicarbonate buffering system, it's intimately related to the respiratory system in the lungs because that's how we move the carbon dioxide around in our bodies and exhale it out of our bodies. So the lungs play a very key role in regulating the bicarbonate buffering system. And to a lesser degree, our kidneys also help us to regulate and control the buffering system in our body. If we need to lower our overall body's pH, our kidneys can excrete bicarbonate ions. We can actually have alkaline urine. It's pretty rare, but it can happen. And then to raise the overall, we can actually, we can have alkaline urine because we're adding an alkaline ion to the urine, lowering our overall blood pH. To raise the blood, I'm going to put a B for blood, to raise the pH of our blood, we will take hydrogen ions out of the blood and excrete them into the urine, and we'll also have the lungs excrete carbon dioxide. And that's how we can regulate blood pH. And we have this physiological range of 7.35 to 7.45. That's the key range we want to stay in. If our pH drops too low, we have an acidosis event. If it raises too high, we have an alkalosis event. And if the blood pH goes below 6.8, we die. If the blood pH goes above 8, we die. We need to maintain this happy medium, the Goldilocks zone for blood pH. Let's look at the phosphate buffering system. As we digest our nucleic acids with catabolic chemical reactions where we break nucleic acids down, we are going to release phosphoric acid into solution. Phosphoric acid is H3PO4 in an aqueous solution. And this is a triprotic acid capable of releasing up to 3 hydrogen ions in solution. I remember when I was taking quantitative analysis, I really despised phosphoric acid and calculating the concentrations of each of these intermediate species in solution. We're not going to do that in this class, thank goodness. And as we look at phosphoric acid, we can have a monoprotic species, we can have a diprotic species, or we can have the triprotic species. And these species will oscillate back and forth. As a diprotic species turns into the monoprotic species, it liberates a hydrogen ion. And in the bicarbonate, just like in the bicarbonate system, this oscillation back and forth is reversible. We have that double arrow in that chemical reaction. So we can go to the left, we can proceed to the left or to the right, depending on whether or not we want to liberate or reduce the amount of hydrogen ions. So with this buffering system, if we are adding more base to our blood, the phosphoric acid buffering system proceeds to the right and will liberate more hydrogen ions to compensate for the addition of more basic chemical compounds into the bloodstream. Conversely, if we're adding more acid to our bloodstream, the phosphoric acid buffering or the phosphate buffering system will move to the left and remove hydrogen ions from solution to compensate for the increase in the amount of acid. So as we look at the phosphate buffering system, this is a very important buffering system in our intracellular fluid. The carbonate buffering system, or bicarbonate buffering system, is very important for blood buffering in our pH. As we look at the intracellular fluid, we have a lot of nucleic acid catabolism within the cell. And that's why the phosphate buffering system is much more important inside of our cells. We have high concentrations of phosphates inside of the cytosol of the cytoplasm. And this buffer inside of our cells is going to function at an optimum pH of approximately 6.8. While it's working, the constant production of these metabolic acids, so we have the pyruvic acid, the phosphoric acid, the fatty acids, we're constantly making acids as a byproduct of our metabolism. So the cytoplasm of the cell tends to be a little bit more acidic than the blood plasma. And as we look at the cytoplasm of the cell will fluctuate from a pH of 4.5 to 7.4, with an average within our intracellular fluid being approximately 7.0 for the pH. If we look at this figure that I included, which is not from your textbook, you can see how the pH in different cells, or organelles I should say, of the cell vary. So a secretory vesicle has a pH of about 5.5, a lysosome is 4.5, mitochondria, depending on where the matrix versus the intramembrane space, could have varying pHs. Remember all the way back when we talked about cellular respiration, how we have with the electron transport chain dumping hydrogen ions into the intermembrane space of the mitochondria. That intermembrane space is more acidic. It has a pH of 7.1 relative to the mitochondrial matrix with a pH of 8.0. So the pH in the cell, generally speaking, is more acidic than the pH of the blood plasma. And to help with that, we are primarily going to rely on the phosphate buffering system on the inside of the cell. That's where the phosphate buffering system is the most significant. We also have a protein buffering system, and this protein buffering system is significant everywhere. We have lots of protein inside of the cell and outside of the cell. So as we look at the proteins in our bodies, they tend to be more concentrated than anything else. Our body, by dry weight, is over 1/2 proteins. And as we look at any protein in our body is going to have amino acids on them. And each individual amino acid is a buffer. So because we have tons and tons of proteins made of tons and tons of buffering monomers, the protein buffering system accounts for the vast majority of all chemical buffering in all of our body fluids, whether it's intra or extracellular fluid. So as we look at our generic amino acid right here at a neutral pH. This generic amino acid is going to have Circled in red, we'll have an amino functional group, and then circled in blue, we have a carboxylic acid functional group. And if we take our generic amino acid and we expose it to an acidic solution, so we add acids, or we are dumping more acids into the system, our pH starts to drop, and the amino functional group has a partial negative charge over here from the lone electron pair on that nitrogen atom. And that lone electron pair on the nitrogen atom will pick up the hydrogen ion from the acid being dumped in solution, and we have the NH2 goes to an NH3. So in response to an acidic solution, amino acids suck up hydrogen ions. And then on the flip side, shift to blue here, if the pH of the solution is rising, that means the concentration of hydrogen ions is going down. The carboxylic acid functional group will liberate one of its hydrogen ions, and then we'll have a negatively charged oxygen as we set up a resonance structure between these two oxygens as the lone electrons, or the electrons, resonate back and forth between the two oxygens. So as we look at proteins, proteins are phenomenal buffers, and they can buffer wherever they are in our bodies. And because they're everywhere in high concentrations, they play a significant role in buffering our bodies. I think I went too far. Let me go back a slide here. Yeah, I wanted to stop on this slide. So we're going to stop here with protein-based buffers. So we talked about the bicarbonate, the phosphate, and the protein-based chemical buffering systems. If you have any questions or comments, please feel free to post them in the class discussion board or to shoot me an email. And as always, happy studies.